Welcome! In this lecture, I will describe the 5 stages of acute animal virus infections. Even though there are a huge variety of life cycles in animal viruses, they all share some common steps to their infections. Your textbook website has some excellent animations to illustrate these stages. Check it out. The first step is always attachment to specific host cells. This is the step which limits which types of cells viruses can infect. In order to attach to a cell, the cell has to have the correct specific receptor on its membrane. For example, the HIV virus uses a glycoprotein found on its envelope to bind to the CD4 receptor that's found on T helper cells. Once the virus attaches to the appropriate host cell, it can then initiate the next stages of infection. Once the virus has attached, it must enter or penetrate the cell. This stage can be accomplished through one of 2 mechanisms. The first is pinocytosis, or receptor-mediated endocytosis. In this mechanism, the virus is essentially engulfed by the host cell membrane in a fashion similar to phagocytosis. The rubella virus enters this way. The second mechanism is fusion, in which the viral envelope fuses with the host cell plasma membrane, releasing the viral particle inside the cell. The chickenpox virus enters cells in this mechanism. The third stage in viral infections is uncoating. In this stage, the viral capsid is removed and the viral genome, not genitals, is exposed. Most families of viruses must uncoat before replication of the viral DNA or RNA, transcription of viral mRNA, and translation of viral proteins can occur. Viruses may use their own enzymes or hijack the host's enzymes to break down the capsid and activate their genetic information. Stage 4 is biosynthesis, or the production of many copies of the viral genetic information and proteins. In this stage, DNA and RNA viruses differ from each other. Because DNA viruses follow the central dogma and have the same type of genetic information as the cell does, these viruses can use the host's enzymes for biosynthesis. For example, the virus can essentially hijack the cell's DNA polymerase to make viral DNA in the nucleus of the cell. The virus can then use the cell's own RNA polymerase to make viral mRNA in the nucleus. This messenger RNA must leave the nucleus and enter the cytoplasm, where cellular ribosomes synthesize viral proteins. Viral proteins, like those of the capsid, must return to the nucleus in order to be assembled into intact viral particles. Clearly, DNA viruses utilize the cell's nucleus. RNA viruses, in contrast, don't follow the central dogma. RNA viruses must carry their own enzymes for biosynthesis, and the replication of viral RNA, as well as all other processes in biosynthesis, must take place in the cytoplasm of the cell. If you remember that the nucleus is designed to protect DNA, it will make sense to you that most RNA viruses don't involve the nucleus in biosynthesis at all. This figure illustrates the generalized life cycle of a typical DNA virus. First, the virus must attach to the host cell. Once attached, it must penetrate that cell and uncoat. Replication, transcription, and assembly of new viral particle takes place within the nucleus of the cell. Eventually, the newly synthesized viral particles are released from the cell, where they can then infect other cells. The nucleus is central to biosynthesis in DNA viruses. As I mentioned earlier, RNA viruses don't follow the central dogma and need to provide some of their own enzymes for biosynthesis. One of these required enzymes is able to synthesize RNA from an RNA template. This enzyme is called RNA-dependent RNA polymerase. This enzyme is different from our own cell's RNA polymerase, which in humans is a DNA-dependent RNA polymerase because it uses DNA as the template for messenger RNA synthesis. Retroviruses, like the HIV virus, have another enzyme known as reverse transcriptase. This enzyme makes DNA from an RNA template, which is essentially the opposite of normal transcription. Single-stranded RNA viruses can be grouped into two categories, the plus and minus strand viruses. Plus-strand viruses are also known as sense-strand viruses because their genome RNA is already an mRNA. The genome can be directly translated into viral proteins within the infected cell. Minus-strand viruses, on the other hand, are known as antisense viruses because their genome RNA must first be transcribed into a complementary messenger RNA before being subsequently translated into viral proteins. Phew! RNA viruses are very complicated. This figure shows the many different pathways of biosynthesis for RNA viruses, depending on whether they're single-stranded, plus strand, single-stranded minus strand, or double-stranded RNA viruses. None of these viruses use the nucleus in their biosynthesis. Again, check out the textbook website animations for a greater understanding of RNA virus life cycles. This figure shows the unique life cycle of the retroviruses, including the HIV virus. After the initial attachment, penetration, and uncoating, the viral RNA is reverse transcribed to make DNA. The viral DNA then is inserted into the host chromosome and becomes a proviral state. From the proviral state, viral messenger RNA is produced and viral proteins are synthesized in the cytoplasm. Viral particles are assembled in the cytoplasm before being released to infect more cells. In review, the first stage of all viral infections is attachment to specific host cell membranes. The second stage is penetration or entry into the host cell. The third stage is removal of the viral capsid and exposure of the viral genome. The fourth stage is biosynthesis, or replication of the viral genome and viral proteins. The final stage, seen here, is the assembly and release of intact viral particles. In this stage, the individual capsomere proteins come together to cover the genetic information. The release of viruses from the infected cell is accomplished through one of two mechanisms, depending on whether the virus is naked or enveloped. Naked viruses wait by the plasma membrane of the host cell and are released as a group or en masse as the cell dies. Envelope viruses are released gradually from the cell through a process known as budding, where the virus steals a piece of the plasma membrane to cover itself with as it exits the infected cell. Check out this animation from the Howard Hughes Medical Institute website to see the amazingly complex life cycle of the dengue virus. When you watch the video, you'll realize that I've just given you the briefest possible outline of the typical acute animal virus infection. This concludes Virology, Part 3.